Well, thank you, Alessandra, and thank you to the organisers for this opportunity. It's great to be here. So I view sort of the problem of muscle aging and muscle disease as a problem of loss of heterogeneity or clonal composition of the stem cell pool. So I'm going to tell you a couple of stories how we think heterogeneity and the understanding of heterogeneity within the stem cell compartment to help teach us about disease and aging. And so as Alessandra just sort of introduced, skeletal muscle is highly regenerative, and this is just a cross-section of an adult mouse muscle. That was the primary model we use. Here is a normal sort of muscle steak. If you inject an intramuscular toxin into the lower leg of that animal, the muscle fibers rapidly degenerate, and you get a robust regenerative response where the muscle looks almost indistinguishable from what it did prior to the injury. At the cellular level, you can see this satellite cell, the muscle stem cell of skeletal muscle, which expresses PAX7, is in a quiescent state, and that's largely because muscle fibers have very low turnover. They're in a very stable state. In response to injury, they break out of their quiescent state, rapidly enter cell cycle, express specific myogenic markers such as MyoD, as you can see here in red. They'll begin to fuse to repair the muscles. They have these central nuclei, which denotes a sort of regenerative event. And then the stem cell pool is replenished, goes back to homeostasis. And this can go on and on and on, indicative of a stem cell population. And through lineage tracing markers, we know that PAX7 cells are both sufficient and required. So if you delete PAX7 cells, you'll have a failure or an abortive regenerative process. So really understanding how these cells function will allow us to understand how disease can be prevented. And these are just the sort of classic lineage tree that we've known about in the myogenic satellite cell lineage for years. My lab over the last few years has sort of realized, as amongst other labs, that this sort of idea of this very simple linear tree is actually way too simple. And so we've been trying to break that complexity down. So conceptually, if you think about heterogeneity, no single cell is really like its neighbors, and how the function of one cell and its preference to self-renew or differentiate, and the numbers of those individual cells will dictate how well a tissue can regenerate or how fast it degenerates. And so the whole premise of my lab is trying to understand this cell from this to this, and how these molecular cues would allow a cell to self-renew, whereas another cell may have a susceptibility to senesce or die. And so we started this project about 12 years ago, based on the idea that across different stem cell compartments, there's an intimate property of quiescence linked to stem cell function. And that sort of preservation of quiescence seemed intimately connected to how well a stem cell could function when prodded. And so we used a genetic approach to test this out, which is obviously a GFP marker, which would allow you to track cellular divisions. So it's a doxycycline inducible element, which turns on GFP within the stem cells. If that doxycycline is removed, every time a cell divides, you'll lose GFP in a predictable and quantifiable manner. So after a period of time, you can see cells that have divided less versus cells that have divided more. And so we use this to track stem cell divisions in the muscle compartment and ask, does that confer a certain functional difference within those heterogeneous population of cells? And so here's the sort of experiment. You can see we use flow cytometry to isolate purified cells. We administer doxycycline to the embryo, and then we chase it out throughout postnatal maturation, where the muscle is increasing about 5-fold, and that is all due to the activity of the stem cells. We isolate the satellite cells, and what we see in the adult muscle, so all of these cells now are satellite cells. Here you've got a population that have divided relatively less frequently than these cells. So these cells hit a quiescent state shortly after birth. These cells carry on dividing and seed the muscle. So not only is proliferative output distinct, we then begin to look at the functionality of these cells. And what you can see here is a transplantation experiment where we isolate the label-retaining cells and the non-label-retaining cells and inject them into transplant of a pre-injured muscle. So this is analogous to sort of hematopoietic stem cell transplantation, where we can inject donor-marked cells into an injured muscle, and those cells will either self-renew or differentiate or won't. And this is a whole map picture of a tibialis anterior muscle from knee to ankle. We've given 3,000 cells. And you can see here there's lots of myonuclei and lots of satellite cells have been derived from this population. In contrast, putting the same number of non-LRCs, these, into the muscle gives a very poor sort of engraftment. And so this was really surprising to us because what it looks like is this is the total satellite cell pool, all PAX7 expressing, and about 30% of the cells are label retaining, possess stem cell activity, whereas about 60% of the population of satellite cells are devoid of stem cell activity. So we think based on this results, we know that sort of stem cell potency is restricted to cells that have a limited proliferative history. And that's just schematized here where we identify that every single muscle fiber has its own stem cell and progenitor. So knowing that you now see two populations of cells, we could think about what happens in disease or aging, rather than losing functionality of all cells, where it seems that half the population doesn't have any, is there a loss of these label retaining cells? And so just to remind you what a regeneration event looks like in an aged muscle, if you poke an adult mouse, you get a nice robust regeneration, new fibers are made, some connective tissue. If you do this to a progressively aged mouse, here this mouse is 32 months of age, so it's almost in the last few days. And you can see there's almost a failure to regenerate the muscle tissue, and instead there's this deposition of connective tissue, fibrosis, and fat. And so real failure of the muscle stem cells to activate and contribute to myogenesis. So we asked a very simple question: are we losing the label retaining cells? And so using the DOX-inducible approach, we chased that out throughout life, quantified the number of LRCs and non-LRCs in an adult compared to an aged mouse of 2 years of age. And so as we've reported and others, you do lose satellite cells. The total population is down by about 50%, but it comes almost exclusively from the label retaining cells. So no matter what population you've lost, it's most important is the functional cells are being lost. We've performed transplantation experiments of these cells, and while they are fewer in number, they do retain potency. So the question is, how can we sort of augment or maintain these or boost actually what would be a label retaining cell, and we're trying to work on that currently. I should also say in muscular dystrophy, you get the same response, where the satellite cell pool is diminished, at least by our methods, and it comes exclusively from losing the label retaining compartment. So this tool has been very useful in the way that it allows us to track cellular behavior, proliferative history, transplantation, but we lose the marker, right? This is green, this is not green. So we're not able to track both of these compartments. So we wanted to take a different approach, and that was to use lineage tracing, which we could use to indelibly mark the more potent or less potent stem cells. And work from our chair of the session, Alessandra, produced a beautiful paper last year where she'd used a PAX7 CRE-ER approach to indelibly mark all the satellite cells. But the nice thing about this approach is they had a different tag and a different fluorescent color on each of the alleles. So you can see this mosaic of colors in regenerating tissue would allow you to just look at the clonal output of each of these colors. And by understanding how big that clone is, you could infer the functional output or the potency of those cells. And what Alessandra demonstrated over doing multiple rounds of injury, which you can imagine is quite a stressful event if you have repeated injuries in the mouse, you get a clonal expansion of a subset of PAX7 satellite cells. I think the unknown question from that paper is what is the identity of these potent cells that can respond and grow under this stressful context? And so we'd been working on this similar question using a sort of orthogonal approach, which was, can we find CRE markers that allow us to selectively mark a more potent cell, and can we track that over time? With the idea if we know the cell, we know its identity, that's the cell that we should target for therapeutic purposes and transplantation for disease. Through a painstaking years of sort of mouse genetics and trying to find different alleles, we stumbled across one, which is MX1 CRE. And so the last session was all about hematopoietic stem cells. So for those in the audience who understand the blood system, we know this is used a lot to mark hematopoietic stem cells because this is an MX1, which is a downstream target of the innate immune system. It's marked in other cell types like pericytes, endothelial cells, MSCs. Basically, this transgene is highly inducible upon type 1 interferon. So if you see a cell that has MX1 CRE, you know it's competent to receive an innate immune system signal. And what we identified when we administered PIPC, which is an agonist of the pathway to drive MX1 CRE induction, we saw a small fraction of satellite cells, about 5 to 10% of the PAX7 cells, which were marked by MX1 CRE. If we put these, and you can see that here in a single muscle fiber co-expressing PAX7 and the MX1, whereas the majority of PAX7 cells don't have that mark. We put those fibers in culture, and we can see they turn on the myogenic program and divide. They express MyoD, they make myotubes and contracting in the dish. So by all intents and purposes, they look quite similar, but they just had MX1. So maybe that wasn't such a relevant thing to chase. But Anna Rita, my postdoc, did a lot more extensive analysis and put these cells in culture and using markers which can distinguish a sort of more primitive versus a more differentiated fate, that being PAX7 and myogenin. Anna Rita observed that the MX1 cells divide in culture but maintain a more primitive state. The more numerous counterparts tend to differentiate. And again, if you want a sort of cell that could be used for transplantation, you want to maintain that self-renewal in that primitive state for a longer period of time. And so we asked, knowing that these were more primitive and our previous work at the label-retaining cells, which were also more primitive, we asked, are MX1 cells a part of the label-retaining cell compartment? And so we crossed mice, we crossed the MX1 tomato with the H2B GFP, we administered the drugs to activate both of the fluorescent alleles, and what you can see here, we get LRCs and non-LRCs. If you ask about the MX1, it's enriched in the LRC compartment. So about 80% of all MX1 cells are in this slow-dividing, limited proliferative output population. And so this is where it gets more complicated. So now we've got 4 different cell populations. We've got label-retaining cells, which have MX1. We've got label-retaining cells that don't have MX1, and vice versa. And so we wanted to ask, who's the best stem cell? And that would be to use a transplantation assay and say, let's see how much muscle you can make. And so there's just some nice pictures to show you that the whole mount, the injection works. Here's a whole mount with the label-retaining cells expressing MX1. So these are all yellow, red and plus green. And these are all green, which are the label-retaining cells devoid of MX1. And we injected 3,000 stem cells, and we quantified the number of donor-derived nuclei. And here you can see by far about 4 to 30% increase in the MX1 positive LRC compared to the MX1 negative LRC. And as we'd previously published, non-LRCs are devoid of stem cell activity. And so it really seems that stem cell potential is restricted to LRCs, and the MX1 positive population is really the one you want that can survive and engraft under the sort of stressful environment of a transplantation. So knowing that we had this sort of potent stem cell, we're pretty excited and asked, how well is this doing in the endogenous context of muscle repair? And this is where things are complicated. So lineage tracing can always have off-target effects, background effects, so it's important you do your negative controls, and we had a nice clean signal. If we administer PIPC, as we see a lot of endothelial cells light up in the muscle, and blood cells. If we perform an injury, here we can see all of these fibers are central nucleated. So we'd injured the muscle extensively, yet we only saw about 5% of those muscles have green or MX1 positive cell contribution. We've tried to injure the muscle again, and again, the rare population of MX1 cells contributed, but with a limited output. So this is really sort of surprising to us that you had this idea of these potent cells under a transplantation context, but these cells were not contributing so much under normal endogenous repair. And so we were sort of scratching our head, and Anna Rita, looking at other cell compartments, came up with the idea that this could be a sort of reserve stem cell population. And so maybe these are not the sort of workhorse of your classic compartment, but really kept in reserves until absolutely essential. And that's been demonstrated very well in the intestine, where stem cells turn over more rapidly, they'll divide and are killed with the radiation, and in that context, a more dormant cell population will recontribute to the whole tissue. So we begin to examine this sort of response of these cells to irradiation, and we saw that in the white bars where the total PAX7 pool is diminished, these MX1 cells are actually rare but preserved. They also have less markers of damage, such as H2AX, and it's quantified here. And so the numbers and the DNA damage response, or the repair, is much more efficient in these cells compared to the more numerous counterparts. If you put those cells in vitro and ask them to grow after irradiation, they maintain their proliferative potential, whereas the others die by apoptosis. Just in the interest of time, okay. So we then begin to transplant these cells in vivo and ask, how do they compare? So we injected the total population of satellite cells, and they were marked by a genetic tag red, and we put the rare MX1 cells at the same number. And this is using live 2-photon confocal microscopy, so we can actually image the same muscle over time. So we put the total population in, and we see engraftment, proliferation, and fusion. and the MX1 cells efficiently engraft, proliferate, differentiate, and fuse in the absence of irradiation. So again, they seem to be a better stem cell. Where it becomes much more marked is after irradiation stress. So here, the total population almost effectively fails. It initially engrafts for 7 days, but it doesn't further proliferate or differentiate. And so now you can imagine your muscle is devoid of new muscle fibers after that irradiation and transplantation. Whereas if you inject those MX1 cells into an irradiated transplantation, they proliferate, they fuse, and make muscle. And that's just quantified here. So there's almost a failure of the total population after irradiation, whereas these cells are almost indistinguishable, at least in the proliferative aspect, compared to the non-irradiated cells. So the final test, sort of like the regeneration, the intestine model would be, do they contribute to endogenous repair after radiation? And so you can think about this for people undergoing radiation or chemo after breast cancer, etc. And so this is just the image of our first injury, very little contribution, but now we irradiate the mouse and we ask how much these cells contribute. And what you can see in the uninjured muscle, again, there's very little contribution. But if you now look at this regenerated mouse, about 90% of all new muscle fibers contributed from this rare population of MX1 cells. If we injure the muscle again to ask, do the stem cells contribute again? They do. And so basically, these cells are essential if you want to regenerate your muscle after radiation. And this is through a clonal expansion and maintaining their proliferative advantage. So we've begun to identify the mechanisms that control the potency of these cells, and through RNA-seq and microarrays, we found one target which we thought was really interesting, and that is PAX3. So for the aficionados in the room, PAX3 has been previously demonstrated to be restricted to a subset of satellite cells. Most thought it turned off during development, and it certainly does, but there are some satellite cells that retain it. And we observed that PAX3 is enriched in the MX1 population, confirmed by RNA here and by RNA scope. Here you can see the puncta in the rare MX1 cells. Now, PAX3 has been demonstrated to be a survival factor, and it's also expressed in melanocytes, and it protects under UV stress there, so we think it's a conserved role. So we knocked out PAX3 now in a PAX3 Cree model. So this is leaving the majority of the stem cell compartment normal, but we can now delete PAX3, expose the cells to radiation, and what we observe is both an increase in DNA damage, marked by olive tail moment, and also increase in cellular ROS. So these cells are sort of DNA is damaged, and they're going through an oxidative stress. Not only do they undergo those detrimental consequences, they all have a propensity now to die through apoptosis, which obviously they were protected against if they had PAX3. So now the final test really is going back to our transplantation model and asking, are we able to contribute to an irradiated injury response if we lack PAX3? And so again, this is a PAX3 Cree ER, and crossed with a PAX3 floxed. So we're either tracking the PAX3 cells or we've deleted PAX3 out of those PAX3 cells. And this just shows you the nice engraftment here from these PAX3 cells. They are potent stem cells. If you lose PAX3 in this transplantation model, which again is stressful, you lose the ability of these cells to respond. Plus IR, seen here, these cells still contribute. And you can compare that quantifiably here to here. There is a small diminution. But remarkably, what happens if you then lose PAX3 in the presence of irradiation, you almost have an aborted ability for these cells to contribute. And so quantifiably here, you can see in the absence of irradiation, but with stress, you get a twofold loss in stem cell potency based on the loss of PAX3, whereas that's about a tenfold loss if you delete PAX3 in that irradiated context. So radiation protection requires PAX3, and PAX3 positive cells are stress resistant. And so putting this into a framework of sort of way stem cell biology has been thought of over the years, we've all been trying to make stem cells self-renew better. And the more self-renewal you get, the better apparently the transplantation. But here's just some recent work from various labs that have targeted different pathways to augment stem cell activity. JAK-STAT, P38 in multiple papers. These are all stress response pathways. And so it may be that actually, while we think about self-renewal, it may be more important to think about the ability of a cell to respond to stress, tolerate that stress. It'll self-renew if it's a stem cell anyway. And so just to finalize with a sort of model, Like all stem cells within a tissue, the satellite cell compartment is composed of diverse clones. And what we've shown is there's LRCs, and there's this subset of reserve cells that are MX1 inducible and PAX3 expressing. Under normal cell turnover, homeostasis and diversity is maintained, suggesting cellular heterogeneity is actually functionally relevant. Under a selective pressure, such as irradiation or aging, we think we get clonal expansion of these reserve cells. And while that might be useful in certain contexts to regenerate that tissue, it could also be a problem if you think about rhabdomyosarcoma or a cell's ability to undergo genetic mutation and not die. So this could be a sort of cell type which is sort of both friend and foe, depending on the context. But I view this sort of stuff in stem cell biology more almost like a Darwinian nature of, it's really about the survival of the fittest. And so we'll wrap up there. I'd just like to acknowledge all the people in the lab who do all the work. The aging stuff was done by a former postdoc who's left the lab now. The MX1 radiation was done by Anna Rita, collaborators along the way, and to the people who fund us to do the work. And thank you for your attention.
Did you investigate the telomere status of your LRC versus NLRC cells when you did the aging study?
We haven't looked at that directly, but we know the proliferative output in those two populations is 4 versus 8 divisions. So I don't think this is a sort of event of sort of replicative aging, but rather I think there's an association of something else that carries along with that proliferative turnover.
PAX3, I mean, there appears to be a relationship between that and HTERT and DNA damage signaling. But
I don't think telomere shortening is the reason why these cells are better or worse.
So what's your hypothesis about the role of PAX3 in somehow tolerating that stress?
Yeah, so we've got a couple of models which we're still trying to tease out. We think that it's definitely playing a role with the chromatin configuration. And so we think there's a sort of maybe protection there. And we don't know if it's a sort of euchromatin versus heterochromatin abundance. That's what we're sort of testing.
Question. So histologically, maybe you mentioned this rare population, is it localized in specific area of the muscle, like close to specific structures that might confer that specificity? Or?
Right, so PAX3 has been known to be expressed in different muscles. So it's expressed highly in the diaphragm, in the pectoralis of the shoulders, and very few in the limb muscles. So if you think about that at the tissue level, you're actually certain muscle groups are going to be able to protect against irradiation a lot more than others. In terms of the cellular localization, we haven't found it. We have looked for it. I mean, we don't see anything close to vasculature. But we think there might be, because of the MX1 link, there might be some immune cells which are close by and they're sort of acting as partners. Okay.
So do these cells just ignore the IR, or do they repair better? Because essentially, if you expose these cells to IR,
yes.
Do they get into the IR-dependent checkpoint, or did you study the repair pathways in these cells?
We've studied the repair pathways and looked at the role of ATM and ATR, and there seems to be a requirement for ATM, but it's very rapid. And so we're seeing things like ATM dependence comes on about the first 10 minutes after IR. So it's very fast. So yes, so I think it's a better repair. And it's rapid. Okay. Thank you.